Okay, so I got that order because point D is the closest to the ground if you were to picture a hill, and point B is farthest from the ground. And we know that potential energy is dependent on the mass of the object, the gravitational constant, and the height, so the distance from that object to Earth's surface. And so in this case, if a ball were to roll around this hill, it would have the highest potential energy at point B because H is at its greatest, whereas with point D, H is at its lowest. Okay, so then for the first thermodynamics question, I gave you 3 situations and asked you to choose the situations that would increase the entropy in a system. And the 2 correct answers are A and B. That is because A shows the transition from a frozen state to a liquid state, so a solid to a liquid. And as we know, the molecules in a liquid are more spread apart. Therefore, they take up more space, and it's a more disorderly state of matter. Then we have B, releasing a constant amount of nitrogen gas from a 4-liter container to a 10-liter container. So the key word here is the constant amount. Because the amount remains constant, we're increasing the amount of space that it's allowed to take up. So as we know, if you were to transfer a gas from one container to another, it would still expand to fill up the entire container, regardless of the size or the shape or the structure of the container. So that is why by giving it more room to spread out, you are increasing the entropy. So then we have use the Gibbs free energy equation to solve the following problems. So problem 2 gives a reaction at 250 degrees Kelvin, where the change in heat is 100 joules and the change in entropy is 0.5 joules. So I probably should have mentioned this, but but when you're doing like Gibbs free energy and stuff, you try and keep the units as kilojoules, not joules. But in this instance, I accidentally had it as joules. So I updated the stream saying that it's supposed to be kilojoules. So if you didn't see that, I do apologize. But yeah, so the answer for number 2 is negative 25 kilojoules. And the answer for number 3 is 25 kilojoules. That was just kind of basic arithmetic using the equation. And then I don't have graphs for you guys, but I figured I can annotate them so we could look over them together. Okay, so imagine that the reactions described in questions 2 and 3 were introduced to catalysts that help the reaction proceed by reducing the amount of activation energy needed. Please draw energy graphs for both situations, labeling, not numerically, the activation energy, initial energy and final energy. Okay, so the way these graphs work are on your x-axis, you have like the progression of the reaction, so I'll just draw an arrow. Then on the y-axis, you have total energy, so I'll do ET, so energy total. And for our first reaction, we have a temperature of 250. The initial reaction is 100 joules. Oh, I'm sorry, guys. And the final amount of energy is negative 25 kilojoules. So it seems like this reaction, because you have a negative amount of like free energy at the end, this reaction is exothermic because it releases energy into the system. So we would have our products with a certain amount of energy, or our reactants with a certain amount of energy, we'll say here. And then of course we have an activation energy, right? So it's gonna take some energy, but because we're releasing energy, our net change should be negative. So this right here is your net change. This would be the delta H. But this is, yeah. Then this right here is your activation energy. And this here would be reactants, and this would be products. So the way this would change for the second problem is instead of having a negative delta G, we would have a positive delta G. So we have our reactants that have a certain amount of energy, and obviously we have the activation energy, and then we have our products, which actually have more energy than the reactants because the reaction took up energy. So this right here would be our delta G, and this right here would be the activation energy to go from, I lied. This would be the activation energy to actually carry out the reaction. Okay, so that's the homework. Does anybody have any questions? Any questions? Okay. All right. So now let's go to the presentation, and I will. Okay, yeah, I can. Great. Okay, so today we're going to talk about enzymes and catalysis. So catalysis is, I guess, the process that enzymes and catalysts use to carry out a reaction. Okay, so let's just get into it. So, okay. So the first thing I want to talk about is an enzyme. So we've talked about these a little bit earlier, but enzymes are specific types of proteins that bind to a substrate. So you're probably wondering what a substrate is. A substrate can be anything. It could be another protein, it could be a molecule, a different type of molecule, it could be a lipid. A variety of things can be substrates, but it's kind of an overarching term to describe something that binds to an enzyme. So enzymes are produced by your body, and they exist to catalyze biochemical reactions. They can break bonds or form bonds depending on the reaction, and they work to deliver ideal conditions for a chemical reaction. So while enzymes are the sites of these reactions, they also serve to alter the pH, temperature, the amount of energy you need in a reaction to make the reaction as thermodynamically and universally favored as possible. So let's talk about the lock and key mechanism. This is the most accurate depiction of the relationship between an enzyme and its substrate. So in this analogy, we'd have the lock being the enzyme and the key being the substrate. So a lock, if you think about something that you might have on your door, it's got a whole bunch of gears within it. And when you put the key in, it changes the structure of the lock so that you can unlock it, right? So enzymes and substrates can do the same thing. Sometimes it'll be the enzyme that changes the substrate. That's the most likely and frequent scenario. But other times we can also have the substrate changing the structure of the enzyme. Okay, so locks and keys are specific to each other. They have a perfect fit. That's exactly the relationship between an enzyme and its specific substrate. So I've said enzyme and substrate a lot, but we can also refer to these as receptors and their ligands. So I'm not sure if I went over this yet. If I did, then you're set. But if I didn't, then I will just I'll just kind of briefly go over it again. But ligands are signaling proteins, and what they do is if your cell wants to talk to a different cell, there's receptors located on the membrane of a cell, and a ligand will bind to that receptor, induce a change, and that change will carry through as like a chain reaction throughout the cell. And the ligand receptor complex has a very similar, I guess, structure situation to the enzyme substrate complex. And both of them relate to the lock and key analogy as they're specific to each other. So you'll have a ligand, and that ligand can bind only to this specific receptor. This specific receptor can bind only to that specific ligand. This is the same situation with a lot of enzyme and substrate There's a lot of different terms for it. Enzyme and substrate complexes, I guess. If you think of like a popular chemical reaction, one that includes a catalyst, then chances are that catalyst has a specific name. So we've named the enzyme something like, usually it's the name of the reaction with A's at the end. So there's no really easy examples for it, but for your homework, you're gonna have to do a little bit of research. So you'll definitely see that. Okay, so when the enzyme and substrate binds, it institutes a change similar to that of a key turning a lock. So just as inserting the key would change the lock structure, inserting a substrate to an enzyme can change the structure as well. Okay. So now talking about catalysis. So for catalysis, we have activation energy. So we discussed activation energy in the previous session, so we're just going to do a bit of a review on it. Activation energy is the amount of energy needed to begin a reaction. So before you're even thinking about completing an endothermic or exothermic reaction, you're going to need energy. Take, for example, the breaking of a bond. So while breaking a bond will release a whole bunch of energy, it is exothermic, it will take a certain amount of energy to break that bond in the first place. So that energy is known as the activation energy. And an enzyme, in the case of bonds, it's referred to as a catalyst, and it can reduce that required amount of energy. So usually the energy will go into setting up the ideal conditions, and an enzyme will work to set up those conditions so that you don't need energy for that, and the energy can be directed at the breaking of the bond. So how do catalysts actually reduce the activation energy? So they can reduce activation energy by providing an environment more conducive to the reaction. One common way is by providing a binding site. This is also known as like the active site. There's a lot of different terms for it. But it basically ensures that when two molecules bind to the binding site, they will come into contact and bond with each other. So if you imagine a glass of liquid filled with the two molecules that are supposed to be reacting, as well as a whole bunch of enzymes, and then you picture another glass filled with only the two molecules that are supposed to be reacting, there's a very high chance that the molecules will come into contact with each other. However, when you have the enzymes present, it increases that chance because you both molecules will be drawn to the enzyme. And let's say it's, obviously this wouldn't be the case, but let's say it's H plus and OH minus, because we know that those come together to form H2O. So there'll be one special place for the H plus and one special place for the OH minus ion. And when both of them come to the catalyst, it will bind them together, ensuring that they actually, that they actually do come into contact with each other. Okay, so ultimately this reduces the amount of energy needed. Usually it'll reduce the amount of energy by changing the temperature. So the energy would be needed in the form of heat, because higher temperatures result in faster moving molecules, so there's a higher chance of collision. This is actually a lot of collision theory that you learn in like AP chemistry. But yeah, so usually an enzyme will change the, reduce the amount of energy needed in the form of heat. So if you had like a really cold environment, but you had enzymes, then a reaction would still be possible. Okay. All right. So here is an image of what would happen if we had a catalyzed versus uncatalyzed reaction. And I do apologize for the graphics, they're not very nice, but it should work. Okay, so the first graph, we have an uncatalyzed reaction with an energy barrier at the top. That's kind of like the max energy that you would need. Then you have the reactants and products, as you know. So we have the same thing in this catalyzed reaction, except one thing you should note is the energy barrier is a lot lower. So the thing that specifically changed is the EA, the activation energy. So what catalysts will do is they'll reduce this activation energy because you don't need as much heat. You don't need anything to change the pH. It's very likely that the molecules will come into contact with each other. So even though the initial energy, final energy stays the same, and the change, the Gibbs free energy stays the same, the EA is all that decreases. And this is good because in the end, it's just more energy for, let's say it's a cell, it's more energy for the cell to use for other purposes. Okay, so here's another graphic that kind of shows the lock and key mechanism fit. So you can see how these two substances would fit pretty well in the enzyme. So it'll enter, then there'll be a little change in shape. So if you look, this is shallow, and this is kind of pointed, right? So now it becomes more curved and this is a lot sharper. So there'll be an induced change, and then the enzyme will alter the products, and then they'll split. So they enter together, and then they're leaving as two different objects, substances. Yeah. Oh, so yeah, they call it an active site, but that would just be the place where the substrate and the enzymes bond. Okay. And then we have one more graphic. This one's a little bit more obvious, like the shape change, as you can see, is very, very clear. All right. So now let's talk about enzyme kinetics. So enzyme kinetics is the study of biochemical reactions that involve enzyme catalysts. This refers to the rate of the reaction being analyzed with varying conditions, notably the presence or absence of the catalysts. So usually in most fields of science, whatever you're studying, you are carrying out experiments, as we know. And a lot of people probably know what the what the scientific method is. But in the scientific method, you have a whole bunch of variables. And your goal is to vary some of these variables. So you'll have independent and dependent variables. But your goal is to vary these variables to see what would happen were things to change. So that's what enzyme kinetics does, except what it's changing is the presence or absence of the catalyst. So one thing you'll get out of this is the reaction rate, which is the frequency of products made as measured by the changing concentration of product. So a whole bunch of chemistry is all about concentration or molarity, which you'll learn in, I think, any chemistry class. But one thing that can be ascertained from knowing the concentration and how it changes is the reaction rate. There's a lot of calculus involved in this, but you don't necessarily need it. I took AP chemistry without calculus. It's just you'll have a lot stronger of an understanding if you know how the calculus behind this works. But yeah, so continuing on, the enzyme kinetics is vital to the understanding of enzyme behavior within the human body because we can know a lot. It's very easy for us to test concentrations within the human body because it's very stable and observable. Can't think of the word, but it's a very stable and observable, I guess, object. Two other variables that you'll get are Vmax, which is the maximum velocity of a reaction, so kind of how fast it's going to proceed, and Km, which is a concentration of a substrate that gives half of the Vmax. So you might know what Vmax is, and you might want to solve for Km. So like what you would do is you would figure out what half of Vmax is, and then find the concentration at that speed. So a reaction will pick up speed and lose speed as it's going. So I want us to think about this. It's a very abstract idea unless you're learning it with all the chemistry knowledge behind it. But if you picture two reactive substances in a container, you have reactant A and reactant B, and then product, we'll call it product C. So reactant A plus reactant B will yield product C. And let's say at the very beginning of the reaction, you don't have any product C. All you have is reactant A and reactant B. So as you release these two reactants to collide with each other, there'll be a lot of collisions at the beginning. And that's because everywhere you turn, you're going to see either reactant A or reactant B, right? It's not going to bump into something that's not one of the two. And the likelihood of it bumping into the opposite is very great. However, as you're producing product C, the amount of reactant A and reactant B is changing, it's decreasing. So it makes the likelihood of a collision, it makes that likelihood decrease. So the speed of the reaction will slow down. It won't be as fast. So there is a kind of sweet spot that gives Vmax, and it's where the concentration of the substrate is at its greatest. Or I have no idea what just came out of my mouth. It's where the, generally, it's where the concentration of reactants are at its greatest. Okay, so let's keep going. Right here, we have a graph, and this graph shows you Vmax, which is kind of like this top line that tapers off. And we have Km, which is the concentration of the reactant, where Vmax is equal to half, or where V is equal to half of Vmax. So if we draw a straight line going up, that's about right here. And we can see that's true. Right here is, so Vi is like half of the velocity and Vmax is the entire velocity. And so what we have here is, and there's a lot of different types of inhibitors, which we'll talk about later, but basically these are different types of enzymes, enzymes that either work, enzymes that partially work, enzymes that don't work at all, and no enzymes. And so you can see what would happen. So this right here is an uncompetitive inhibitor. So an uncompetitive inhibitor shouldn't really change Vmax, but we can see that it is making it a little bit more difficult. Then we have a non-competitive inhibitor, which results in quick growth at the beginning, but it kind of slows down, and it slows down a lot. Then we have a competitive inhibitor, and then a normal enzyme. I guess it's pretty intuitive. You can study this graph a little bit later if you'd like in more detail. But yeah, let's keep going. So these are the different types of inhibition. So we have competitive versus non-competitive inhibition. Competitive inhibition occurs when you have an inhibitor, which can be like any kind of molecule, and it binds to the active site of an enzyme. So by doing that, it's blocking the possible binding of a substrate. It's kind of like going to park your car in your parking spot and realizing that there's another car parked there. You can't do anything, right? And this ensures that the reaction will in no way proceed because the substrate can't bind anywhere else on the enzyme besides the active site. And if that active site is blocked, then there's just no chance. So then we have non-competitive inhibition. So this occurs when you have an inhibitor that doesn't necessarily bind to the active site, but it binds to a different place on the enzyme. So what happens is the structure of the enzyme will be changed slightly due to the binding of the inhibitor, and it won't be able to function at its most efficient level. However, the substrate can still bind. It would just be kind of weird. Perhaps you get a different product, or maybe the substrate would have to be aligned in a certain way for it to bind. Either way, it'll reduce the likelihood of substrates binding and the reaction successfully occurring. So here you can see a normal reaction, which is where this right here is your active site. The substrate will bind, something will occur, and the products will be released. So here we have a competitive inhibitor. This is the purple molecule is your inhibitor, and now the substrate cannot bind. And then we have a non-competitive inhibitor. So the inhibitor will bind somewhere else onto the enzyme, and the substrate can bind, but the reaction just won't proceed normally. And that's because the binding of the inhibitor will change something within the enzyme that messes it up. Okay. So here we have allosteric regulation. So allosteric activators are very similar to non-competitive inhibition, except in this case, they're activating. So a molecule, let's say you have an enzyme, and the enzyme doesn't usually work. Generally, it's a dud. If you have a certain molecule, the activator, it will bind to a separate site on the enzyme and induce a structural change. The structural change will increase the enzyme's affinity for substrate and activate it and make the substrates bind to the enzyme and something to occur. Then we have feedback inhibition. So this is a very cool type of inhibition and super, super common in your body and outside of your body. So it's a type of inhibition that can be either competitive or non-competitive depending on the situation. Molecules of this product will bind to the enzyme, changing its structure and either blocking the substrate or decreasing the affinity for the substrate. So the catalysis is entirely dependent on the amount of product already present. I think I have, perfect. Okay, so right here, if we start here, we have a substrate. It binds to the enzyme, and you have another substrate, it binds to the enzyme, you have another substrate, it binds to the enzyme, and then eventually you get this end product, okay? This end product will go all the way back to the first enzyme, this thing right here. It'll bind to it like a non-competitive inhibitor, and now your substrate can't bind to it anymore. So it's kind of like a feedback loop. So when enough product is made and enough products bind to the enzymes, then the reaction will stop. Proceeding. So another type of inhibition is phosphorylation, and this actually is different because it occurs on DNA. So DNA, if you don't want to transcribe it, the transcription of DNA requires the use of enzymes. It's very similar to enzymatic activity. And the way it's regulated is through phosphorylation. So it's a form of inhibition that involves the addition of phosphate groups. These phosphate groups are commonly seen in the blockage of DNA transcription and can be used to inhibit enzyme activity. So binding phosphate groups will change the structure of the DNA, the structure of an enzyme, in the same way that any other inhibitor would, and it just prevents transcription or catalysis from occurring. Okay, so any questions? Okay. Then that is all. Thank you, and I will see you next week. There should be a homework, or I'll have a homework assignment up by tonight. So, yeah. Oh, wait.